Okay, and just check. Oh, I'm still screen sharing. Okay. All right. All right, everyone. Hello, guys. I'm going to record these videos for you guys as I won't be here tomorrow. So in this video, I'm going to talk about what we should have talked about today and the contents of tomorrow. I won't make it overly, I won't make it overwhelmingly a lot. It's a terrible amount of contents that you need to review, but just the essentials, right, that are on your notes. Again, you have your notes in the OneNote online, and I'll be printing out the PDF of this once I add more notes to it, so I won't have to redoing the PDF. Okay, so we're gonna go over and continue the different organelles of the cell. If you remember from the last class, I was talking about the Golgi apparatus and lysosomes and the ER and all the different organelles from the cell. And we're almost finished. There's a couple of cell structures that I want to talk about that are not visually included in the book, but I did want to show you guys how it looks like and its overall function. It is included in the table, but they don't really mention it more in detail. So let's begin off. So I'm going to talk about cell membranes. There's an entire new chapter that is just about cell membranes and how it works, which is chapter 9, that we're also going to get into in this video. So for now, I just want to talk about that the cell membranes are relatively thin layers, right, that serve as a boundary from the internal contents of the cell to the outside environment. And I'm going to go into that more detailed in the next chapter. But if you look at this micrograph here, you notice that compared to the overall cell, that these organelles, this structure is pretty thin compared to the overall girth of the cell. So take a look here. It's less than 5 nanometers, right? But you can still kind of see the double layer that we're going to go over the next section there, right? So the cell membrane essentially regulates the material that enter and leave the cell. It provides the boundary for the cell. And though there are material that can't readily get inside the cell, so it needs to undergo a certain process that is called endocytosis, which you guys are familiar with. Basically, a lot of molecules get into a certain region of the cell, and the cell engulfs it by wrapping around a little bit of membrane. And then it turns into essentially a vesicle, right, that will go into the cell area, right? And the opposite of endocytosis is exocytosis, where the vesicles that are made from the ER and from the Golgi apparatus, they're being liberated along with the contents inside these vesicles to the outside via this process, right? Exocytosis. All the content gets spilled out of the into the extracellular material. So that's regarding membranes for now. The next organelle I want to talk about are centrosomes and centrioles. Now, this is not in your book. It is talked about in your book, but it doesn't show how it actually looks like, especially on a electron microscope image. But essentially, these are tubular structures. Okay, they come in pairs, right? There are these tube structures, right? You see a bunch of tubes that they're organized in a particular way. And when you have these tube structures together, they're called the centrosome. Okay, but each individual one is referred to as centrioles. And they're just a collection of tiny microfilaments, microtubules, and they serve, moreover, in cell division. They elongate the cell, they contract the cell. They also help with the cell skeleton, but moreover, these particular structures, they help for cell division. Okay. Usually they're located at one area or one side of the cell. If you take a look at this diagram here, you see how it's geared more toward this lower side or that base side of the cell. they're usually in the extremes, right, of the cell. Um, you do have another one that is closer to the nucleus, especially during cell division. They kind of divide to the extremes of where the cell pulls each other. But we'll take a look into that in units 3 and 4 more in detail. Okay. And Yeah, they essentially serve or function primarily during cell division. Okay, you can see a micrograph here of these centrioles and centrosomes. Here you would see one of them, and here you see another one. But they're usually in pairs, right? So this all together is the centrosome, and you can see a bunch of little filaments being shown here that are the little microtubules throughout these little centrosomes there. Okay. The next couple of structures that I want to talk about, you're familiar with. They're regarding the plant cell. Okay, so you know some of the structures that are specific to the plant cell, which is the cell wall and the chloroplast. This is very, very familiar to you already. The cell wall being a thicker layer surrounding the cell membrane, which is primarily made of cellulose. Okay, and they help essentially protect the wall and keep the cell wall, the plant cell itself, kind of rigid and give support to the cell. Okay, giving that overall structure, texture, shape to the wall, to the cell. Not just to plants, but some bacteria and fungi also have cell walls. The material might be a little bit different, but they have a similar function of cell support, right? Plants are made out of cellulose. Fungi are made, and some bacteria made out of chitin, depending how you want to say it, right? Another structure that the cell has is the central vacuole, all right? This big open space that you see here. And it essentially serves to basically store water and nutrients. It could also store waste, toxic waste products made from the metabolism, but other foreign kind of toxins from the environment there, right? And they do help give overall rigidity to the cell, right? Because they do have a lot of water. And whenever a cell gets dry, is a plant cell gets dry, is because the water is being removed and basically evaporating, and it's kind of, you're dehydrating the cells, right? So the cells start to wilt, and the plant starts to wilt overall. So you always want plants to have a large vacuole filled with water, right? Okay, so here we've talked about the cell wall, we've talked about the central vacuole, right? In this electron micrograph of plant cells, you can see them readily here, right? You have your chloroplast, which I won't go over because I already did in the previous structures. All right. But one of the more defining features or largest features is the central vacuole in terms of plants, right? And you can see that kind of in the diagram there, giving that idea. Okay. So finishing off, you have this whole idea of all the organelles working together. Ultimately, you have a system that's referred to as the endomembrane system. And this whole system is essentially to produce proteins, to assemble them and package them to wherever they need to go. Ultimately, it starts with the nucleus, right? If you see step number 1 here, proteins, they're getting the building instructions for from the DNA. And if you remember from last year, term 1, we talked about RNA replication, RNA synthesis, DNA synthesis, transcription, translation. That's essentially what's going on here. You will not learn that right now, but we will see that in the future, right? So ultimately, everything comes from the nucleus. The information of the DNA passes down to the ER and ribosomes to start manufacturing the proteins, right? These proteins, they're first into amino acid chains, and these acid chains get further modified and modified, and they get transported, right, in this whole endomembrane system to the Golgi apparatus, right? Always via vesicles, right? So if you see vesicles, the ER does produce vesicles, but these guys tend to go straight to the Golgi apparatus, right? Um, and these vesicles, they join the Golgi, and the Golgi finishes off modifying the protein and creating uh, more kind of intricacies with the protein, right? Labeling and reshaping the protein as much as it needs before it's finally repackaged again in another vesicle and sent outside the cell. Or it could be inside the cell, depending on where it needs to go, right? But usually it's outside the cell via exocytosis, right? So these diagrams here are a kind of summary. I do recommend that you study these diagrams. I didn't go over every single bullet point, but it's because we've already kind of reiterated that. I have another form of a diagram here so you can get the more visuals you see and the better you understand, right? So you can interpret what's going on here, right? But it's similar ideas there, different forms of imagery, but as long as you get that same concept. Information from the nucleus goes to the ER, or rough ER, I want to say. Rough ER, then the protein that is being assembled goes to the Golgi, and from the Golgi, it goes and is excreted outwards. Okay, so take a look at these diagrams here to get like an overall summary of what the endomembrane system is, right? But that's essentially the organelles that you need to know. All right. Any more than that, I will let you know. But I think these are the essential ones that you need to memorize. Okay. I've also placed on your notes an overview, right? A little chart overview of what you need to learn. Okay, here's a nice colorful diagram of the animal cell, each with its points. I strongly recommend you take a look at that. You don't need to know peroxisomes, we haven't talked about. Microvilli and the whole cytoskeleton system, you don't need to know that, right? And flagellum, for now, you don't need to know that, okay? And chromatin, at least for now, you don't need to know that. I will explain to you that in more detail. All right, so you have the animal cell and you have the plant cell, which you guys can take a look better. There are some examples here of unicellular eukaryotic cells, okay? Like this type of green algae here, to give you a good sense of how different organisms look like, not just multicellular cells, but unicellular organisms also. Okay, so again, take your time to kind of analyze this table also and these diagrams also. All right. So the actual class that we're supposed to talk about today is the cell membrane, right? So that's why I didn't talk about it in a lot of detail. And some specifics that you need to know regarding cell membranes and transport processes on how the membrane regulates materials coming in and out is all involved in chapter 9, right? So some kind of questions that we might be asking ourselves within chapter 9 is, how do cells obtain their requirements, right? How are they going into and collecting material that they need and removing things that they don't need from their body outside, right? How do cells respond to the environment to keep them always in homeostasis, right? For example, at high salt concentration or low salt concentration, right? We're going to be talking about the fluid mosaic model, which is the most accepted model right now. They're the latest model on how the cell membrane works, right? Understanding how things work by modeling is essentially one of the key things about science, right? You can't directly see everything through the microscope, but you need to get some type of interpretation on how it might work, right? Based on evidence, okay? So yeah, some of the things we're going to learn today are the cell membrane fluid mosaic model, transport of materials through various ways in the cell membrane, by active or passive processes, and ultimately how size affects this idea of materials processing and moving in and out of the cell, right? Okay, so I have placed here the part of the pages for you to have here, but you could get it also from the website, right? But I'll be pasting all of the book pages here for you to have. Okay, so let's talk about section 1.9, which is the cell membrane. All right, one of the key things that they're going to talk about that they always mention in the book and many other resources is the terminology, right? Cell membrane is also called cellular membrane. It's also called the plasma membrane. It's also called being selectively permeable membrane, right? They're all essentially the same thing. Now, the idea of selectively permeable, I will talk about that in a little bit more in detail. All right, but essentially, what is the overall function? For everything that we talk about in the cell, you need to understand there's a relationship between the structure and the function, right? What does it do? What is its purpose? And why is it that the way that it's built is particular to its function, right? So in this case, you have a cell membrane, which is a very thin layer, a bilayer more specifically, that helps serve as a boundary between the internal environment of the cell and the external outside environment, right? The internal contents and the outside environment. Some things, some general functions that we're going to be talking about is of the cell membrane is what it does, right? The functions, which include, recognizing other cells nearby and seeing, hey, you're my same cell type, you're my bro, or you're a different cell type, you're a bacteria, right? You are definitely not related to me, and you're actually a danger to us. How do we, go on about that, right? Aside from that, transportation of materials through the membrane, right? To and from the contents of the cell, the inside of the cell, and what are some ways that molecules can attach to these cells, right, for a particular signaling pathway to occur? It could be by enzymes, it could be hormones, it could be other nerve cells, the little tips of nerve cells that are touching actually the cell membrane, right? So by having physical and chemical properties, the cell membrane is able to serve as a pretty good boundary for the cell, right? And we're going to talk about some of those things a little bit more in detail. Okay, so I do want to mention, so some organisms do have a cell wall, like I just mentioned before. That is not the same thing, and I will repeat that because some people tend to get confused. The cell wall is not the same as the cell membrane. The cell wall covers the cell membrane, right? It's a lot thicker and harder and less flexible. Okay, I do want to mention, if you notice here, I put CM. I will be kind of annotating cell membrane as CM just for simplicity's sake, for you guys to get that idea, right? Shorter vocabulary, but you guys should know already. CM means cell membrane, right? So the cell wall basically adds strength and support for the cell membrane and the cell overall, right? But the cell membrane is the one responsible for selecting what will come in and out through the cell. Okay, that idea of selection is referred to as being selectively permeable. Okay, choosing by receptors, by transport proteins, one way or another, choosing what exactly will come in and how much and at what times, right? Is called being selectively permeable. An example of the cell membrane, a nice diagram, is found here. If you were to basically enlarge a cell as much as you can and just look at the covering, all right, you would see this kind of double layered membrane that you see here. These two layers, whoops, these two little layers. Oh no, I lost myself, guys. Sorry about that. There you go. These two little layers here that here you can see one layer going down, All right, then you have that other layer. And we're going to talk about what it is composed of. But this is covering the entire cell, right? It's its boundary, right? Um, it's its covering. If you were to get a little segment of this cell, let's say this little box that you see here, and you take it out and we look at a little side view of it, it'll look something like this, right? A cell membrane is actually composed of a lot of little sections, right? Some little units that are referred to, if I take out one individual one, it's referred to as a phospholipid. This here you must memorize, all right? You need to know every single thing. Phospholipid. That is terrible handwriting. All right, phospholipid. All right, and why is it called a phospholipid? So the reason why is because at the top part of this complex molecule, this little purple area that actually looks like a sphere, it has a phosphate group. That's why it's called a phospho and then a lipid, right? And these phosphates and these glycerols, this is another molecule that belongs to this little head, right? You usually just think about a phosphate, but it also has a little bit more. It has a little glycerol group. You don't need to know the chemical formula for this. You don't need to memorize, but you do need to know that there's two parts to the head, and it's the phosphate and the glycerol. And they refer to this little head part of a phospholipid as being hydrophilic. And the reason why is because it has a lot of oxygen atoms that give this head a kind of charge, a semi-charged value, right? Positive or negative. It has a charge, right? And because it has a charge, it's said to be polar. Okay. Molecules that tend to have oxygen and any elements from that group of the periodic table tend to have a charge, right? Slightly negative charge. All right. They're referred to as being polar. At the other extreme of this structure, these little tails of the phospholipid, they're referred to as a hydrophobic tails. And they are made up of a lipid structures that more specifically are called fatty acids. You do need to know these terms. There's two types of fatty acids based on the chemical composition, right? If you remember from physical science, you have elements, carbons that have double bonds, you have carbons that have single bonds. You don't need to know that in general, but just know that every time you see these little double lines parallel to each other, they're referred to as unsaturated fatty acids. Okay, unsaturated fatty acids. If you don't have any double bonds between carbon molecules, they're referred to as saturated fatty acids. Now, the thing about these molecules is that they generally, or for the most part, don't have oxygen. They might have a little bit at the end, but they don't have that much oxygen. It's just carbon and hydrogen bonded to a chain, right? That is represented by these little tails. Okay, now I'm going a little bit too much into detail, but I just want you to know that these are lipid molecules, and lipids don't tend to have a charge. So they're referred to as being nonpolar. Okay, so you have a polar head that likes to face the water because water itself is a polar molecule. And then you have the tails that are nonpolar, right? And nonpolar molecules are usually lipids like fats and oils, right? So because of this nature of the phospholipids, they tend to organize themselves in these little arrangements. Okay, you have one phospholipid in which you have water on this side, and you also have a kind of water on this side. So all the phospholipids, they reorganize themselves so that the tails face each other. That way, the tails get away from water. Okay, they fear water. That's why it's called hydrophobic, water fearing. As a result of that, through many, many molecular interactions throughout the years, evolution, you have these structures of the cell that are bilayers of phospholipids. So that is why, more specifically, the cell membrane is called the phospholipid, okay? Bi meaning two, layer, okay? Bilayer. So you have two layers of phospholipids that is referred to as the bilayer there. So in general, for this little term, I want you guys to know cell membranes, they have a structure that enables them to carry out many functions that we've talked about before due to this ability of creating a bilayer, okay? Now, I do want to talk about some properties that the book does not mention that you need to know, though, for the WACE program. And for that, I'm going to show you a little demonstration, okay? And that'll be in for this first video, okay? I'm going to show you guys my little screen. I hope this works, that you guys can see this. Am I really, let's see. Stop sharing here. I'm still recording. All right. Good. Alt. All right. So I'm going to share again my screen, but now I'm going to share with you my camera. All right, so let me go over here. I want to show you guys a little demo that I wanted to show you in the classroom, but I think here is equally fine. I have this set up here. You guys can take a look. All right. This is my elaborate cell membrane kind of setup here that Ishikawa-san helped me make, right? So what I have in this case is a model representing some parts of the cell membrane. All right. So if you see here, these little circles indicate the phosphate lipids. The areas in red, these little lines in red, are the tails, right? That they're inside the membrane. Now, what I've done here is added these little balls that represent the phospholipid heads. But what I want you to focus on is more over how the balls move, right? So the cell membrane actually is embedded with several materials and things. But moreover, before we get started on that, I do want to mention a couple of things. The cell membrane is very flexible, okay? And do we have orange color? Sorry, guys. Because I'm gonna do a little bit more drawing here. But one thing that I want you to understand is that the cell membrane isn't just a plastic layer that, you know, is rigid, it's hard. No, it's very fluid-like. The layer itself is very fluid, like water. And one of the reasons why you get this kind of rigidity and fluidity is that it has little structures that is called cholesterol, right? Now you can't see it so much here. There's little cholesterol molecules within, or maybe you could see it a little bit, within the cell membrane that gives it fluidity. And what I mean by fluidity is that the actual cell membrane could bend and wiggle, move around like the surface of the water. So what you're gonna see here is these little balls that are going to simulate how the cell membrane actually moves around, right? So if I do this, right, the cell membrane very nicely, it's not hard like a piece of wood. Um, it it tends to flow and move like a wave, just like that, right? And of course, the bottom part of the little sticker they taped on the wall. But what I want you to understand is that every little phospholipid, you know, kind of moves in a very wave-like motion, right? It's flexible. And if you were to press down on the cell membrane, you know, the little phospholipid heads can actually, you know, they can withstand a little bit of force, like these little balls here, right? And they can reorganize themselves. All right. Aside from just the phospholipid, the cell membrane also has structures embedded like little icebergs right? These structures are proteins, right? And proteins could float on the top part of a membrane. Let's say like this particular protein here, and it could also move. So let's say um, uh, on this top layer of the protein, the proteins can actually move around, right? Getting closer to this area, moving around over here. There's some proteins that stay just on the top side of the cell membrane or on the bottom side. I'm designating the bottom side here, but actually, technically, the aluminum should be right here, right? But for simplicity's sake, guys, let's say this ball here is the whole membrane, right? They stay only on one side of the membrane. There's some membranes that actually pierce through the entire and I'm going to move it closer here so you guys can see. They move through the entire transmembrane, okay, both bilayers, okay, and you can see a little bit of the membrane here at the bottom inside the cell, and a little bit of the protein at the outside of the cell, the external membrane, right? These are called transmembrane proteins that they pierce through this whole middle portion of the cell membrane. Now, one thing that's actually super interesting is that the phospholipids, and I'm going to move my little protein out of the way for a second. The phospholipids in the cell membrane is not fixed. All right. So if you notice right now, the phospholipids themselves, they are bobbing up and down, right? But actually, they can even move from different locations. And what do I mean by that? I'm going to move the screen a little bit and take a look at this orange little phospholipid. All right. If this is the top part of the cell membrane, okay, right, you're seeing the top part of the phospholipids. The phospholipids themselves can move around. Let's say this little phospholipid is going around here and they're changing direction all the time, right? So now this little orange phospholipid, they're sliding past each other, okay? And so forth as you go along more and more. So the initial position of this phospholipid, let's say, was over here, right? It can move around just because the cell membrane is so fluid and flexible that they're able to move into like a sea of phospholipids, right? And within that sea of phospholipids, you have little iceberg proteins that are embedded in there, right? Some of them above the phospholipid, just like this protein, or some of them under the phospholipid bilayer inside the cell, like the one below. All right. So I did want to mention that idea. This whole concept that we understand of how the cell membrane works, right? Where you have phospholipids moving around, that they're fluid, that they act like a fluid, and there's proteins embedded on the cell membrane, right? With other molecules. This is called the fluid mosaic model of the cell. All right. You must understand this. This is one of the more critical things that you need to understand. Okay. Um, uh, in terms of cell biology, right? It's one of the key points for you guys to know. All right. So make sure you understand that part there. Okay. So let's continue back. I did talk about this already. Now, I want to go over actually discussing more over the cell membrane. So I won't go over all of the notes here today. I just these two, maybe just this notes, and that'll be good for today. All right. Some more examples of the cell membrane, right? Like I mentioned before, the cell membrane is very, very flexible, right? As you saw that it could move around in a wave-like motion. It it can actually reshape the overall organism, right? This is especially important in unicellular freshwater organisms like the amoeba that you see here in this picture here, right? It can alter its shape. It can alter the composition of the cell membrane, the shape and size, depending on what it needs to do. Let's say like feed, right? To give you a sense of what I mean by that, take a look at this little video here, right? You don't need to know the audio here really quick, but what I want you to see is how nicely, oops, this is like terrible zooming in. What did I do here? Let me redo that. Let me put that in HD here. That was 1080. Let's get it up. So you see how the cell membrane is extending actually, right? As the amoeba is moving along, as it's kind of creating temporary arms, extensions to try to trap a food particle there, right? Another organism. Cytoplasm and membrane is shifting from different locations to another because of that same idea, right? That the phospholipid bilayer can even move around and different molecules can go from left to right to alter itself to a different shape. Okay. Essentially, the amoeba in this case will engulf the organism there. All right. So that's why I mean that the cell membrane is quite flexible. Okay. Um, so I've been talking about how the cell membrane kind of adapts and is able to uh, regulate the internal contents and external environment. But we're constantly exposed to the outside environment, right? Exchanging substances between these different areas, right? The external environment now has a particular name, a more specific name. It's called extracellular fluid, right? That I'm going to be referring to as ECF. So be familiar with these terms. And the extracellular fluid is basically everything, every liquid that's outside of the cell, right? It's further categorized into other things, but in general, it's called extracellular fluid. The inside stuff that's inside the cell, right? Basically, the cytosol with the dissolved particles and everything is referred to as the intracellular fluid, ICF. So you have ECF and ICF there, right? The intracellular fluid and the extracellular fluid. Okay, and the intracellular fluid is basically, the water in the cell contents, the ions that are dissolved, and so forth, so forth. So if you take a look at this little diagram here, so I'm going to designate this in these notes to this picture here, right? You have your cell, right? And the stuff inside the cell is called the ICF, right? Everything that's outside of that cell is referred to as the extracellular fluid, and it's broken down into two further things. The interstitial fluid, basically the liquid that is in between two cells, right? And you have some fluid that is in blood vessels for multicellular organism that is called plasma, right? Ultimately, for you guys, all you need to know is extracellular fluid, right? Because it does get a little bit more in depth. Okay. And the reason why we need to kind of regulate what's outside and what's inside is quite interesting. And the book gives a nice analogy between humans and Earth. And if you were to think of the human as just one single mass, right? The majority of humans, the majority of the content of humans and other living organisms for that matter, is that 99% of our mass is made up of just these 6 elements: carbon, oxygen, hydrogen, nitrogen, calcium, and phosphorus, right? We do have some other ones, but they're not as great as these in terms of quantity, right? We're 99% composed of these elements here. And what's quite interesting is that when you compare, and I mentioned this to you in the last term, right, when we were doing kind of a brief overview, if you notice, among each individual element, carbon takes 18% of the proportion in the human body, right? Only 18%. You would think carbon would have more because it's the one that has the most um, uh, skeleton for all the molecules, which is true. If you were to compare that to the percent of carbon on Earth, it's only 0.03%, which is very interesting. We're quite opposite in terms of if we were to compare ourselves as structures, right? We're quite opposite in terms of percentages, relative percentages. Oxygen actually takes the cake with 65% of our composition, right? Not just as a gas form, but also as a solid form. Different compounds can create different structures. Doesn't have to be a liquid, doesn't have to be a solid. Take a look at H2O, which is they're both hydrogens, but together they make a liquid. Carbon, when it bonds to oxygen, creates many, many different organic compounds. Okay, so we're 65% oxygen. The percent on Earth is 47% of that. Um, on Earth, percentage in seawater is 86%, and in the atmosphere, it is 21%, right? So different relative abundances. Hydrogen, if you take a look, we're 10% made up of hydrogen, right? And so forth. Nitrogen, 3% only, our body. But very, very interestingly, in the atmosphere, take a look, nitrogen is 78%, a staggering 78% in the atmosphere. So a lot of it's pretty much lost. We have to kind of adapt to it in the nitrogen cycle. Okay. So one of the things that's important about kind of like regulating the extracellular fluid with your internal, with your intracellular fluid and content is that it's always changing, right? The environment's always changing. So a problem with unicellular organisms is that they're always in constant touch with the external environment since they're just one cell. For example, an algae that's in pond water, right? It's always needing to regulate its salt content into and out of the cell to keep it stable, right? But imagine if suddenly that pond gets very salty for maybe too much runoff, too much erosion happening, that there's a lot of salt in there. The algae needs to quickly be able to sustain that difference in salt concentration, or else it will die, right? So the cool thing about multicellular organisms is that the majority of our cells are surrounded by this fluid, which is pretty constant, right? It has a pretty constant salt concentration, pretty constant pH concentration. So we don't have that problem of suffering, being exposed to changing environments, right? We have evolved our skin to kind of protect ourselves to do that, and other organs to protect ourselves from too much salt, from too much pH, right? Which is basically the lungs, the kidneys, the liver, that helps us regulate that stuff pretty well. Okay. If you take a look at this table here, and I'm going to kind of designate these little arrows for you guys to get a sense of what I'm talking about here. Take a look at this table, and it's pretty much the difference in concentration between extracellular and intracellular fluid, right? Sodium in the outside has a concentration of 145 millimole per liter, and the inside is 15 millimole per liter. Potassium has the exact opposite, almost the exact opposite. Chloride has 116 in the outside environment, right? The inside is only 20. And calcium has more on the inside than the outside. So there's some instances where we cannot be balanced. We actually need a difference in concentration. An example of this, that we need a difference in concentration, is actually sending nerve signals through the body. And more particular, we use sodium and potassium ions, right? You do not need to know this for now, but I just want you to understand like why the body does that, right? We need to maintain concentrations because certain actions or certain things require this, like moving a nerve signal. A nerve signal is basically an electrical impulse due to a difference in ion concentration. So I want to show you this really quick. And why is it that this is possible? Is because the cell is selectively permeable. Now take a look at this. You have sodium ions, which are positive ions, and you have potassium ions, which are also positive. So how can two positive ions make a negative charge? Take a look at the quantity. Look here, you have 1, 2, 3, 4, 5, 6, 7. You have 1, 2, 3, 4, 5. What does that tell you? All right, this is positive, 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 positive. Here you only have positive, 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 positive. So just by definition that this one is more positive creates this to be a little bit more negative, right? They're both positive charges, yes, but because this one has more content of positivity, if I can say, this one by default ends up being less positive, so it starts to get a slight negative value because there's just less positive charges there, all right? And that's how an electrical impulse starts being created there. As the cell membrane begins to open its channels to create differences in potential, take a look what happens now. It opens its gates for potassium by ATP. It's forcing more positive charges into this area, giving a negative net potential. And then you have a change. Potassium is also changing now, and sodium and so forth. So it's like a little lever system that you're changing negative, positive, negative, positive. And that change ultimately sends a signal throughout the nerve, right? That's a little bit too much detail for you guys to know, but understand the reason why we need to create concentration changes, right? For example, to make a nerve signal, right? So the cell has devised, evolved different ways to do that, right? And not just for sodium and potassium charges. Different cells need different amount of things for different purposes. For example, high cells that need to produce a lot of protein, right? They need a high level of amino acids, the building blocks of protein. So it's kind of logical, right? If you're going to produce a lot of fiber, let's say in your muscles, you need a lot of amino acids to produce that fiber. Let's say your nerve cells, like I just talked about, nerve cells depend on ions to send a signal, right? So they're going to need a lot of ions in these particular cells. Do you think skin cells need a lot of ions there? Most likely not, right? Because they serve for protection and not for nerve signaling. Okay. Ultimately, what I want you to know is that cells always regulate their content. All right. From the inside contents of their cell to the outside contents of their environment, right? So yeah, so this is what I want you guys to go over. This was essentially today's class and the class that I'll be absent on Thursday, right? Okay. I'll explain this more in detail in the next class. I think it's good for us to go over it together. That's it for today, guys. Thank you for watching this video at any time at your convenience. Try to have this by Friday so we can continue on more in detail. All right. You can pause it, fast forward it if you want. If some things are familiar already to you, then you can do as you wish, right? But make sure you go over, especially the demonstration, that stuff is not in the book. Okay. With that being said, guys, that's it for today. And we are finished for the content.